Good morning, everyone. Today I'm presenting our recent work on enhancing the vaccine names re-ranking using the large language models. Yeah, this work has been done with Dr. He's group and Dr. Tao's group. So the background, as we know, the vaccines have revolutionized the public health, and it provides protection against infectious diseases and cancers. Clinical trials is a valuable repository contains the clinical trial information. It evaluates the vaccine performance. And also it contains a lot of vaccine name information, but those vaccine name information lacks standardization. And also there's challenges in automatic concept mapping, evidence-based decision-making, and vaccine surveillance. So our goal was to try to map the vaccine names in clinical trials to the vaccine ontology. So the vaccine ontology is a community ontology developed by Dr. Hu, and it promotes vaccine data standardization, integration, and computer-assisted reasoning. So here, this is our motivation. The vaccine name mapping is crucial for standardizing the clinical data. Ontologies like the vaccine ontology provide a structured framework for organizing the vaccine-related data. However, there are some challenges like the variability in the vaccine names. Since in clinical trials, the vaccine names may be inconsistent. The same vaccine name may have different various format. Also, there are insufficient contextual information. We have done some preliminary work. Those preliminary studies show that there are much space for improvement for the top one accuracy. Yeah, here is our objective. We try to improve the accuracy and efficiency of mapping vaccine names from clinical trials to standardize the vaccine ontology concept. We try to leverage large language models to enhance the re-ranking of vaccine name mappings. This is our preliminary work. We have developed a cascaded framework to mapping the vaccine names in clinical trial to vaccine ontology. We use this strategy like fine-tune the multiple domain specific language models for mapping VO from CT. And we also applied weighted rule-based ensembles to enhance the mapping performance. And our preliminary work also supplemented the concept of auto-coverage in current vaccine ontology to enrich its vocabulary and further enhance its interoperability with other ontologies. This is the cascaded framework. As we can see, firstly, we try to pre-train a language model based on POMAD and clinical trial text. Then we have a CT POMAD. We further fine-tune this CT POMAD plus the sideboard and on the humorous corpus. So we got a CT POMAD plus sideboard fine-tune the model. To better align with our mapping goal, so we further fine-tune this CT POMAD plus sideboard with the vaccine ontology knowledge and the clinical trials vaccine names information. Then finally, we get the CT POMAD plus sideboard plus VO fine-tune the model. And finally, we utilize a weighted ensemble approach to try to map the vaccine names from the clinical trial to the vaccine ontology. This is the preliminary work evaluation results. We can see that although we applied a series of domain-specific models, fine-tuned models, the performance, especially for the top one accurate performance, still is not too much high, right? So as we can see, the highest performance is our finally fine-tuned CT POMAD plus the sideboard plus VO. It achieved 31.8% for the top one accuracy. However, the top 10 accuracy we can see is 81.8. this is the bottom rows or the ensemble results. From the ensemble result, you can see we finally improved the top one performance using the ensemble re-ranking ensemble method to 62.7%, and the top 10 accuracy achieved 87.3%. Then we may have to think about how to improve the top one accuracy, since as we know from the top 10 accuracy, it achieved 87%, but the top 1% is only 32%. If we can apply some models, especially like the large language models, to re-ranking the top 10 candidates, then we may further improve the top one accuracy. So this is our follow-up work onto re-ranking the initial mapping results, try to improve the top one accuracy. Yeah, this is the procedure, like the data collection and preprocessing, gather comprehensive dataset of vaccine names from clinical trials, pre-process the vaccine names to remove inconsistency, such as abbreviations, typographical errors, and variation in naming conventions. And then we did the initial mapping. We use the three baseline ensemble models, the score-based, the scale-based, the ranking-based, for initial mapping from preliminary study. Then we applied the large language-based re-ranking. We employed GPT-4, Google Rank, and Google's MedLM-Medium and MedLM-Large for ranking initial mappings. And finally, we do some evaluation using the top 1 to top 10 accuracy matrix. Yeah, this is the large language models we utilized in this study. The GPT-4, Google's MedLM-Medium and Large. Yeah, Google's MedLM-Medium and Large is specialized in medical domains, tuned to understand and generate medical text. The Google's rank is another model for ranking. So we just try to find how about this ranking model, the performance on this re-ranking of vaccine names task. This is the dataset we utilize in this study. We utilize the vaccine ontology and the ACD database. The ACD database is just a reorganized relational database from the original clinical trials data. So it's just try to store the original clinical trials files into a relational database. We also utilize the VDOS2024 release for fine-tuning those domain-specific models. This is the data processing and screening. As we can see first, the ACD database, we identified 745,000 records. And those records, there are many is long license that will be removed, have been removed. And finally, we get 6,472 records screened. And together with this, further extracted vaccine names from the clinical trial based on the vaccine ontology knowledge. So finally, we achieved to get a cohort of 7,873 records included in this study. So this is the re-ranking method. As you can see, firstly, we control the vaccine names corpus, then do the initial mapping by ensembling cascaded fine-tuned domain-specific large language models. Then we utilize the large language models to do the re-ranking using the prompt tuning. Then finally, we get the evaluation results. The experimental results showed that GPT-4, the performance is the best result, achieved the best result. It achieved 80.2% increase in top one accuracy over the baseline models. And the MedLM models showed the performance similar to original ensemble model. The Google rank exhibit a lower performance with their top one accuracy only 40%. So this is the comparison of the top 1 to top 10 accuracy for all the models. We can see that GPT-4 achieved the best performance from top 1 to top 8, top 9, top 10, all the models are the same results. Since these 4 neural network models are based on the original results, the top 10 results. So the top 10 should be the same. And the top line also is the same. So from this study, we can see that the neural network models, particularly for GPT-4, significantly improve the accuracy of vaccine name mapping. And integrated neural network models can offer a scalable and efficient solution for biomedical test normalization. So this is the future direction. We'll continue to explore hybrid approaches combining neural network models and traditional models. And we also try to further fine-tune models with domain-specific training. And also, we are trying to integrate contextual information to enhance the prompt tuning for neural network models. So in conclusion, mapping and re-ranking vaccine names from clinical trials to ontology concept is very challenging due to the variability in vaccine names and insufficient contextual information. Utilize neural network models like GPT-4, Google Rank, MedLM-Medium and Large to re-rank the initial mappings. And to incorporate additional contextual information to enhance prompt tuning for neural models can further improve the re-ranking. So this study highlights the promising role of neural models, particularly GPT-4, in enhancing vaccine name mapping and re-ranking, setting the stage for further advancements in biomedical text normalization. Yeah, so thanks for Dr. Ho's group and Dr. Tao's group. This work have been done with the cooperation between the 2 groups. Thank you.
Excellent. Thank you very much, Dr. Lee, for great talk. So we have 6 minutes for questions. Everyone's quiet. All right. Maybe I can start with a few questions. Just for clarification, when you first, evaluated the multiple different methods and ensemble approaches, you had an accuracy. Can you explain how you actually calculate those accuracy?
Oh yeah, this accuracy is just for example, this you mean this temper or yeah, both or is okay. This is the initial result. This is the final re-ranking results. The top 1 accuracy is that, for example, the original result was achieved with this is the score ensemble method. Top 10 results. For example, the model try to map the vaccine name from clinical trial to the background, the vaccine ontology. Since there are many vaccine names in the ontology with thousands of, then the model try to compare each candidate in the vaccine ontology to the query vaccine name, then calculate the similarity, then rank it, and finally it extracted the top 10 highest similarity as the top 10 candidates. Then, for example, for top 1, the mapped the original vaccine names to the top 1 vaccine name in vaccine ontology. But this mapping may be correct or not correct. So it will calculate the accuracy for all the queried vaccine names, the dataset, then averages, then there is this is the so-called top 1 accuracy. For the top 10 accuracy, you will do for top 10 accuracy, then there may be, for example, the query is only 1, but the candidate will have 10, right? So you will compare all the top 10, whether the top 10, at least if there is at least 1 candidate is the corrected mapping, then it is flagged as a corrected mapping, or then there is a missing mapping, something like that. Then finally, also average all the query vaccine names results to calculate the top 10 accuracy.
Yeah. Thank you. I think so. What you did is so you identify those potential mapping and check whether these identified terms are indeed in one of those vaccine ontology term out of 1,000, couple of thousand terms.
Yeah. In fact, if, for example, in top one, for the top one, I kind of try to map to compare the similarity between the query vaccine names to thousands of vaccine ontology vaccine names, then ranked them based on the similarity score.
Mm.
Then it extracted the top 10 as the top 10 our results.
Yeah, and the similarity, when you say the kind of match, is the kind of perfect match, like score, similar to score 1.
Yeah, similarity is just calculated. For example, there are different similarity method or algorithm, and then we just rank those scores, then get the top 10, then calculate the accuracy for each top, like top 1, top 2, top 10, top 1. If is the, for example, if the vaccine, the query vaccine name is mapped to the correct candidates in the vaccine ontology, then it's flagged as true, or it's flagged as false. Then for all the test vaccine names, it's just average them, then get the final top 1 accuracy. Yeah, top 10 is the same, similar procedure, yeah, to calculate.
Okay. Thank you.
Yes, thanks.
Anyone else does anyone have other questions? Otherwise, I have one more question for Dr. Lee. So when you fine-tune the model with in the UMLS data set, you use the whole in 5 gigabytes of data. But vaccine ontology or vaccine terms are only a small fraction of the UMLS. I wonder whether you could try only those vaccine relevant component of UMLS to fine-tune. And I don't know, do you think that might have different results in the outcome?
Here, this is just try to say UMLS contains medical domain knowledge, right? So we just try to utilize the medical domain knowledge in the UMLS corpus, and to see sideboard is a fine-tuned ranking model.
Yeah, I understand. The question is, because the UMLS is kind of about the whole medical domain knowledge is, but since your work is focused on the vaccine ontology or vaccine area, I was wondering whether using a subset of knowledge on vaccine from UMLS might have different results.
Yeah, this is a good question. But yeah, since UMLS is a general medical domain, it contains many knowledge about it other than the vaccine, but it also contains a lot of vaccine information, something like that. In here, this sideboard is just this sideboard ranking model is initially fine-tuned on the UMLS based on the original POMAD board. But here, we just try, since we already pre-trained a fine-tuned CT POMAD board. So we just replace the original sideboard is the baseline model from the POMAD board to the CT POMAD board, then to fine-tune it again. Since here, this UMLS is not our further, in fact, is not our fine-tuned domain knowledge.
I see.
It's for the sideboard's own domain knowledge. We just try to incorporate the CT knowledge, so to fine-tune again, so to get the CT POMAD sideboard. Yeah.
Okay. All right. Thanks for the clarification.
Yeah.
Does anyone have any other questions for Dr. Lee? Otherwise, let's thank the speaker and move on to the next one. Thank you, Dr. Lee.
Thank you.